Hello, my name is Molly, and today I will be giving a brief to help better understand the COVID-19 vaccine. I want to start this off by saying that the intention of this brief is not to pressure anyone to get the vaccines. This brief is going to be talking about those things that need to be understood before we can actually even start talking about the vaccine itself. Let's start diving in. First of all, hi, who am I? Why am I giving this brief? Before I joined the Navy, I was working in the Yale School of Medicine in the Department of Computational Biology and Bioinformatics, and our research was primarily focused in the field of genomics, which is the study of all of the genes in the human body, collectively called the genome. And then I was also working on specifically some human genome privacy projects. Before that, I worked at Pfizer in their drug discovery group, and particularly with the high throughput sequencing folks. So that involved dealing with a lot of data, publishing protocols, managing a lot of compounds and data and protocols was primarily what my role was while I was at Pfizer. Before that, I was getting my master's in molecular biosciences and bioengineering. I was in a proteomics lab. As I said, with genomics, it's the study of the collection of all of the genes in the human body, which is the genome. And proteomics is the study of the collection of all of the proteins in the human body. And then we were taking proteomics and we were applying it to a lung cancer problem. Proteomics, as it turns out, was a really valuable field for me to have studied because a lot of the immunology relies on the interaction of proteins. Specifically, how the coronavirus hijacks our human cells relies on a protein-protein interaction between the spike protein and the ACE2 protein. We'll go into all of this, so it's okay if it doesn't make sense yet. Before that, I was getting my Bachelor of Science in Mathematics, and I got a minor in biology and a minor in anthropology, and it was that minor in biology that really inspired me to go on this path through the biological sciences before I joined the Navy. As part of my bachelor's, I was an undergraduate teaching assistant for a cell biology course. And in that cell biology course, they learned the foundations of all of the organelles in the cells. They learned how genetic information is turned into proteins and then how proteins do things in the cells. In my master's, I was a graduate teaching assistant where I walked them through an experiment and then we had them write a paper on it piece by piece. We taught them how to communicate scientifically with their peers, which is also a very valuable skill. So I give you my credentials, not to say that I'm an expert by any means in any of these fields, but rather that I have enough of a foundational knowledge and I have enough experience to be able to read the scientific literature, digest it, and disseminate it to a wide range of audiences. By the end of this brief, you all can expect to learn some of the impacts of COVID-19 in the United States and worldwide and why a vaccine is necessary. We're going to talk about adaptive immunity, what it is, how it works, and the roles that vaccines play in developing that adaptive immunity. We're going to talk about the typical vaccine development timeline, and we're going to compare it to the Pfizer timeline. Next, we're going to talk about how the coronavirus specifically hijacks our human cells and turns them into virus-making factories. Then we're going to talk about the types of vaccines that are available and then what is in messenger RNA vaccines specifically. We're going to go over the Pfizer vaccine safety and efficacy. Then we're going to go over some common myths surrounding the COVID-19 vaccine. Lastly, we're going to talk about how to sort through COVID-19 vaccine misinformation when you come across it on the internet or out in the world. So first of all, why do we care? Well, as of March 18th, 2021, there were over 29 million COVID-19 cases in the United States. There were over 130 million worldwide, and all of these cases resulted in over 500,000 deaths in the United States and over 2.23 million in the world. These cases have also resulted in literal trillions of dollars being spent by the United States alone on COVID relief programs. There were also a lot of jobs that were lost as a result of this pandemic. Just in one single month in 2020, there were over 20 million jobs lost in the United States. In Asia, that number was around 80 million. Worldwide, it was unknown, but just with the United States and Asia, in those two regions, we lost over 100 million jobs, which severely impacted our economies. I'm sure, as all of you are familiar with, we also had to experience lots of travel restrictions and mandatory quarantine periods. People have had to quarantine for 14 days whenever they do have to travel. We've also had to isolate a lot, and with quarantining and isolation, we've experienced a lot of loneliness, and loneliness without a pandemic is already one of the greatest killers in the United States and worldwide. But especially with this pandemic, we have seen substantial increases in both suicide ideation and deaths by suicide, particularly among young adults and teenagers. And with all of these jobs that were lost, and as economies have slowed or come to a complete stop in some countries, we have experienced a global recession that rivals that of 2008. So it's really important that future COVID-19 cases be prevented. And while we can wash our hands and we can socially distance and we can wear masks, medically, the only treatment that provides preemptive protection from viral infections is vaccines. But in order to talk about vaccines, we first have to talk about how adaptive immunity works from a very high level. So it starts off with a macrophage. And the macrophage, it's a white blood cell. And its job as a white blood cell is to patrol the human body looking for something that causes disease. For example, a virus. The more generic term that we use for something that causes disease is a pathogen. And what happens is the macrophage, it patrols the body looking for a pathogen. And when it comes into contact with it, it doesn't know what to do right away. So it picks up the pathogen and it breaks it down into its protein components. And then it presents those protein components to a second type of white blood cell called a T lymphocyte or a T cell. Now, this T cell, it's going to do one of two things. The first thing that it can do is it can turn into what we call a killer cell or a killer T cell. Killer cells or killer T cells, what they do is they patrol the body looking for human cells, and they look for human cells that have been infected with the pathogen. And the way that they can tell that the cell has been infected with the pathogen is because the proteins that the T lymphocyte were presented with are on the pathogen that are coming out of the cell. So it recognizes those proteins coming out of the cell on the pathogen and it destroys that cell. Then the remnants of that destruction are digested by the macrophage, that first type of white blood cell. So that's the first path that this T lymphocyte can take. The second path that it can take is going on to activate something called a B lymphocyte or a B cell. B lymphocytes, they're responsible for making these little Y-shaped guys called antibodies. Antibodies are a very specific defense mechanism against the protein that was presented to the T lymphocyte. The tips of these antibodies, the tips of the Y shape, they will change their shape depending on what the shape of the protein is. So I'm going to do a demonstration here. If the protein has a shape that looks like this, the antibody, the tip of it, is going to have a shape that's perfectly complementary to it so that it can bind very tightly to the protein on the pathogen. And that very tight binding is crucial because of two things. The first is, let's suppose that the protein on the pathogen is responsible for binding to your cells to hijack your cells and then turn them into virus-making factories. If the antibody is binding to that binding site on the protein on the pathogen, it's blocking that binding site, and now that binding site can't be used to bind to your cells to hijack them. So that's the first potential benefit if the antibodies are targeted to that specific binding site. The other thing is when enough of these antibodies are sticking to the protein on the pathogen, the tail ends of these antibodies are made of sugars, and that signals to the macrophage, that first type of white blood cell, to come and digest the pathogen that's been covered with antibodies. So that's a really powerful way that our bodies can fight off pathogens, or in this case, a viral infection. But the thing is, to get that perfectly complementary shape to the protein that was presented to the T lymphocyte, it can take a long time. It's something on the order of 10 to the 8 different combinations that the B lymphocyte has to go through before it gets those antibodies that are perfectly complementary to it. Once it figures it out, it's going to create these plasma cells that produce those specific antibodies. And on top of that, our bodies want to remember how they created that perfect shape. Our body makes something called memory cells, which retain the information to make that specific shaped antibody that's complementary to the binding site of the protein that was on the pathogen, which was then presented to the T lymphocyte. The reason for that is so that if you get infected with the same virus that has the same proteins on it, your body can respond to that threat right away. And that's really powerful. A couple of things about adaptive immunity. The first exposure response can be very slow. As I said, we have something on the order of 10 to the 8 different combinations for those tips of the antibodies to get them to fit perfectly with the protein on the pathogen. It can take 14 days, for example. And during that time, whether you have symptoms or not, you are going to have a weakened immune system and you could be contagious. Sometimes when you have symptoms, they can be pretty severe. They can result in the death of a patient. If the symptoms of the virus directly cause your death, it is considered the primary cause of death. But there is another way that viral infections can result in death, and this is what we call a comorbidity. It's a secondary cause of death. So what this would look like is, let's suppose a patient has COVID-19. They have a weakened immune system while they have COVID-19, whether they have symptoms or not. While they are sick with COVID-19, they come into contact with bacterial pneumonia. And under normal circumstances, they would have been able to fight off that bacterial pneumonia. But because of their weakened immune system, they die from it. In that case, COVID-19 is considered a comorbidity. So when you're looking at the cause of death section of a death certificate, there are 2 boxes. And the first is the primary cause of death. That's this box right here. If a patient had COVID-19, but they died from bacterial pneumonia, then this first box would be talking about the bacterial pneumonia. They would include the COVID-19 disease in the secondary or this contributing cause of death. And these types of deaths are counted as COVID deaths. After you survive a viral infection, you have those memory cells, and those memory cells make the antibodies that are perfectly tailored to that protein on that pathogen. What's valuable about that is the next time you are exposed to the virus, your body's going to recognize the virus very quickly, and it's going to be able to shut down the virus before the virus has the opportunity to replicate in your body. And there are a couple of reasons why we don't want the virus to replicate in your body. The first is that when it can replicate, that means you're going to be carrying more virus inside of you at a given time. We call the amount of virus that you have in your body your viral load. And depending on what your viral load is, if it's high enough, then you are considered contagious. Now, the viral load is going to vary depending on the virus that you're dealing with. Some viruses are more infectious than others, and so you might need a lower viral load to pass on a more infectious disease, and you'll need a higher viral load to pass on a less infectious disease. So when the body shuts down the virus before it has a chance to replicate, that means that you are far less likely to become contagious, which is very important for the next reason. When the virus is not allowed to replicate, that means the chance of it mutating is going to be lower. So when it has the opportunity to replicate not only in your body, but in the bodies of the people who you pass it on to, statistically, more replications equals more opportunities to mutate and have that be a mutation that affects our ability to fight off the viral infection. So, for example, if the protein that our T cell was presented with looks like this, and we developed antibodies that are perfectly complementary and bind to it, we are relying on that protein to stay in the same shape. But if the virus has enough opportunity to replicate, there's a chance that at some point a mutation will occur that changes the shape of that protein from something like this to maybe something like this. And so you have antibodies that are perfectly complementary to this shape, but are not perfectly complementary to this shape. So now you can't bind to the new protein, which means that you are dealing with a new strain of virus to which you are not immune. We want to make sure that people get exposure to the virus, but we don't want them to deal with the full-blown infection that comes with first exposure to the virus. This is where vaccines come in. Vaccines are aiming to trigger the adaptive immune response by providing that first exposure without weakened immune systems and contagion that comes along with the organic exposure. When a, quote, large enough percentage of the population has developed immunity to a virus, either via vaccine exposure or via organic exposure, we term this herd immunity. The percentage of people who need to be immune to achieve herd immunity, it's going to vary by disease. For measles, which is a highly infectious disease, the percentage of people who need to be immune to it to achieve herd immunity is 95%. But for polio, which is less infectious, you need only approximately 80% of the population immune to it. When it comes to COVID-19, we hear a lot of different numbers in the media saying, maybe you need 70% immunity, maybe you need 95% immunity. And there's really not a very clear answer. A lot of people can guess, they have educated guesses based off of other coronavirus infections. But the real answer is that we're not going to know what the threshold is for herd immunity for the novel coronavirus until more people are immune to it. So the way that it's going to work is we're going to look at cities that have an immunity level or a vaccination level of 60%. We're going to look at that city, we're going to look at that population with 60% immunity, and we're going to see, do we see spikes occurring? Do we see spikes in infections happening? Do we see an increase in the number of cases there? If so, then we can say, okay, looks like 60% is not really enough to achieve herd immunity. Then we're going to look at another population where the vaccine rate or the level of immunity is about 70%. And we're going to look at it and say, are we seeing increases in the number of COVID cases, or are we not? Are we seeing spikes in the number of COVID cases? If we are, we're going to look at another city. And we're just going to keep looking at different numbers until we get a good approximation of how many people, what percentage of the population needs to be immune to achieve herd immunity. Why do we even bother defining a threshold for herd immunity? What really is important about herd immunity? Not everyone can receive a vaccine, and not everyone can risk organic exposure to a virus. There are people who are immunocompromised, such as organ transplant recipients. There are patients who are cancer patients or who had cancer in the past who had to undergo chemo radiation therapy. There are children who are under the age of 2 who cannot receive vaccines yet, or certain vaccines. There are HIV and AIDS patients. There are a whole host of reasons why people cannot receive a vaccine or why they cannot risk exposure to the virus, because they will very likely come down with the disease and die from the disease. One of our goals with vaccines is to provide immunity to the herd so that they can surround the people who are vulnerable and protect those people from exposure to the virus. Now we're going to talk about a typical vaccine development timeline. So normally, vaccines can take anywhere from 10 to 15 years to 20 years to develop. And the way that we normally do things is we start with the research, which is what do you put in your vaccine that'll make it protect people? Then there's some preclinical work that needs to be done. For example, maybe animal trials, mouse trials. Then they go into phase 1 human trials, followed by phase 2, followed by phase 3. And after the phase 1, 2, and 3 trials are complete, then there's the building of factories, the manufacturing, the approval, and the distribution. Now, with the COVID-19 vaccine timeline, we didn't skip any of these steps, but we were able to save some time because we did some of them at the same time or with some overlap. Now, none of this can happen until this first step is done, which is what do you put in the vaccine that will make it protect people? So to answer that question, we have to understand how the virus actually works. So we're going to talk about that. So this is a picture of the coronavirus, and it looks like a ball with a bunch of spikes on it. And some of these spikes are bigger. We call those S for spike protein. And then we've got these little bumps on it. These are called M proteins, M for membrane. Then we have the viral envelope, and inside of the viral envelope, we have the squiggly blue stuff, and the squiggly blue stuff is the messenger RNA. Messenger RNA is genetic information, and it's the instructions to make the spike protein, the membrane proteins, the viral envelope, and more messenger RNA. So what happens is the spike protein on the coronavirus, it finds a surface protein on one of your cells, this big blue blob that is one of your human cells. And on this human cell, there's a surface protein. It's a receptor called the ACE2 protein. The ACE2 protein is found pretty commonly on lung cells and on the cells lining the airways. And so it makes sense that this particular virus, one of the symptoms of the disease it causes is respiratory distress, because it specifically targets cells that have this ACE2 protein on them, which is primarily your lung cells. The spike protein, it binds to the ACE2 protein. And when it binds to the ACE2 protein, it sends a signal to the cell to bring the virus inside of it. And once it brings the virus inside of it, the virus opens up and releases that genetic material. So the genetic material, some of it goes on to get copied over and over and over again by your cell's machinery. But some of it goes down into a part of your cell called the endoplasmic reticulum, or the ER for short. In the ER, particularly in the rough ER, there are ribosomes. The ribosome is a protein complex that has the job of reading the messenger RNA instructions and turning them into each of the protein components of the virus. The messenger RNA goes to the ER, the endoplasmic reticulum, and the endoplasmic reticulum gets to work on making the spike protein, the membrane protein, the viral envelope, etc. And then once you have all of the pieces of the virus, the next step is to assemble them. And that assembly happens in what's called the Golgi body. The Golgi body assembles the viral proteins together along with some of the messenger RNA. And once it's all assembled, the complete virus is sent out of the cell via a process called exocytosis, and the virus goes on to infect another cell. But this messenger RNA that's left behind, it gets copied over and over and over again. So it gets copied, sent down to the ER. The ER makes the spike protein, the membrane proteins, the viral envelope, and it all gets packaged together in the Golgi body, and then it makes a new virus and it sends it out. Your cell has effectively been hijacked into making virus. As the cell is releasing the virus, you have killer T cells who have been trained to spot maybe the spike protein or maybe the membrane protein. So they see this cell releasing virus. Killer T cells will find this cell and they will kill it. Now remember, this is one of your cells that's involved in your airways. This is one of your lung cells. It could be lining the alveoli, which is where the oxygen exchange takes place between the blood and the lungs. So if your killer T cells are destroying your lung cells because they've been hijacked, you're going to suffer from difficulty breathing. That's one of the reasons why respiratory distress is one of the biggest symptoms of COVID-19 infection. Now that we know how our enemy works, we're going to talk about the types of vaccines we have available in our arsenal. The first is called live attenuated. This is where you get a small amount or a weakened version of the live virus. Now, the good news with these live attenuated vaccines is that they provide a very strong response by the immune system, and so you have long-lasting immunity to the virus. The downside is you are getting whole virus. These vaccines are risky because, in theory, if your immune system isn't able to shut down the virus before it has a chance to replicate in your body, then you could develop the full-blown infection that is caused by this virus. This was not going to be the type of vaccine that was going to work for this massive vaccine campaign. The other thing that we have is an inactivated vaccine, which is where you get the whole virus, but it's been deactivated or, quote, killed. Killed is in quotes here because viruses are not technically alive. They lack a metabolism and they lack any machinery for their own replication. They rely on your cell's machinery to do it. But we can deactivate them. The downside to these vaccines, while they are safer, is that you might need to get several booster shots with this type of vaccine in order to maintain that immunity. Next, we have subunit vaccines, which is instead of getting the whole virus or a small amount of the virus, you're getting a piece of the virus. And what's great about these types of vaccines is that there is zero chance that you are going to become contagious. The downside to these vaccines are that you have to make that piece of the virus in a factory in these big tanks in these cells. And so it can take a long time and it can be very difficult to do and it can be very expensive. But we also have another type of vaccine called mRNA vaccines. These are relatively new vaccines where instead of making the piece of the virus, instead of making the subunit in these big factories in these tanks of cells, the mRNA vaccines give the patient the instructions to make the spike. Instead of it getting made in a factory, it gets made in your body. But just the spike, the protein that binds to the ACE2 protein on your cell to get inside of your cell and hijack it. Active ingredient in the Pfizer-BioNTech vaccine is messenger RNA. Messenger RNA is genetic material that will, in this case, encode the viral spike protein of the coronavirus. Messenger RNA, it only codes for the spike, not the membrane proteins, not the viral envelope. Why would they bother to put anything else in this vaccine? They needed to in order for this vaccine to actually work. So messenger RNA is the key ingredient. It's the active ingredient in this vaccine. But messenger RNA is something that we call hydrophilic. It loves water. It loves being surrounded by water. Now your cells, they are surrounded by what's called a plasma membrane. It's the skin of the cells, and it's very oily. And so we call it hydrophobic because it doesn't love water. It's water fearing. Now, messenger RNA, being something that loves water, can't get across the oily membrane of your cells that hate water and don't want to be surrounded by water. So the question is, how do you get something that loves water across a membrane that hates water? In order to get the messenger RNA inside of the cell, we're going to use lipid nanoparticles. And lipid nanoparticles are made up of lipids. Lipids are things like fats and oils that are naturally produced by our body. They have these really long names, but that's just so that chemists can be very precise when describing these different lipids. So the way that this works is you've got a sphere of oil and you open it up and inside you put the messenger RNA. Then this lipid nanoparticle is going to merge with the cellular membrane. And when it merges with the cellular membrane, it's going to release the messenger RNA inside of the cell. So the purpose of the lipid nanoparticle is to transport the messenger RNA across the plasma membrane of the cells. Next, we have pH buffers and salts. pH refers to how acidic or how basic a solution is. So if you were to get hit in the face with a very strong acid or a very strong base, your face is going to melt off essentially, or it's going to burn. And so lipids are very susceptible to changes in pH and changes to acidity or how basic a solution is. And so we put these buffers in there that will prevent the solution from becoming more acidic or more basic than we want it to be. Next, there are salts, which also aid in making the vaccine more comfortable to administer. Last, we've got a stabilizer, which is sucrose, and this is used for when the vaccine is repeatedly frozen and thawed. The sucrose helps to stabilize it during that transition. So a couple of things. The first is that it doesn't contain any mercury, thimerosal, or preservatives. The reason why this creator decided to include this in their slide is because there is a debate on the internet, because the internet is rampant with misinformation. There is a debate that vaccines cause autism because vaccines contain mercury, thimerosal, which is some very high percentage made out of mercury, or because these vaccines have preservatives in them. And regardless of where you stand on that particular issue, this vaccine doesn't contain any of that. So the discussion is moot there. There is nothing in here that has ever been argued to cause autism. Further, vaccines do not cause autism. but that's a debate for a different day. Vaccines do not cause autism. This vaccine doesn't contain any of those ingredients. It didn't need any of these. Now that we know what's in the vaccine, we're going to talk about the Pfizer-BioNTech safety and efficacy. So we're going to go over a couple of different things. First, we're going to talk about the safety. So to understand the safety, you have to understand what sorts of things they were looking for in these vaccine trials. First of all, there were over 37,000 participants in this trial. And the way that vaccine trials work is they take half of the participants, and they give them the actual messenger RNA vaccine. That's going to be this column on the left. The other half, they give something called a placebo. The placebo is essentially a shot that has nothing in it. The placebo is given to people because of a couple of reasons. The first is we want to know what percentage of people can you expect to experience certain symptoms, whether they got the vaccine or not. For example, let's suppose that you get a vaccine and then you go and eat at a restaurant, and then later that night, you're feeling some sort of gastrointestinal condition. Maybe you have heartburn, or maybe you are vomiting. You don't know if it was from the vaccine or if it was from the restaurant where you ate. So the reason why we have the placebo is to see if these people who get the shot, if they experience certain symptoms, even though they never actually received the vaccine. We want to know how many people we can expect over the course of some period of time to experience certain symptoms that are in line with some of the symptoms that are experienced by the people who did receive the vaccine. So that's the first thing, is we want to know how many people we can expect will experience a given symptom over a period of time, even though they didn't receive the vaccine. The second thing is that there is a real effect in medicine called the placebo effect. And the way that it works is that if your brain believes that something that you are receiving is going to have an effect on you, positive or negative, sometimes your brain will actually manifest that symptom or that benefit in a measurable way. For example, if your brain believes that the vaccine is going to give you a fever and you receive the placebo not knowing that you got the placebo, your brain might cause your body to develop a fever, and you can measure that fever, and that is amazing. We use the placebo in science to determine how likely it is that a given symptom is actually due to the drug or the vaccine that we're testing versus how often a particular symptom occurs naturally in a population, even though they didn't receive the vaccine. So the way that this works, we're going to go through these first three rows, is Pfizer looked at what is called an immediate unsolicited adverse event, AE, within 30 minutes after vaccination. So what this means is that they gave 18,801 people the first dose of the vaccine, and they didn't ask them anything. They didn't solicit a response from them. They just told them to go and sit down and wait for 30 minutes. And out of that roughly 18,801, it was 0.4% who came over to the administrators and said that they experienced some kind of symptom, some kind of adverse event. But in the placebo group, which was similar in size, 18,785, it was also 0.4%. To me, what this looks like is, and I don't know if this is the case, but the way that I interpreted this when I was gearing up to get my vaccine is, let's suppose that I am volunteering to participate in this trial. I don't know if I'm getting the actual vaccine or if I'm getting the placebo. Further, the person who is giving me the vaccine can't tell me if I'm getting the vaccine or the placebo. Let's say I don't know much about vaccines, and I particularly don't know anything about this vaccine. Is my heart rate going to increase? Am I going to maybe have a queasy stomach, maybe feel like I have to go to the bathroom, or maybe I'll feel a little bit of lightheadedness? These are all symptoms of anxiety. And so these people who received the placebo, they received a shot that had nothing in it, but they didn't know if they were the shot that had nothing in it. And so whether you get the shot or not, you can experience that, or you can expect that people who get a needle injected into their arm, about 0.4% of them are going to experience some sort of symptom within that first 30 minutes after vaccination. Now, after dose 2, it was 0.3% of people who got the actual vaccine who reported something in the first 30 minutes, and the placebo group had 0.2%. So yes, this 0.3% is technically a little bit higher than this 0.2%, but statistically the difference between 0.3% and 0.2% of a combined over 36,000 people, this is statistically what we would call not a significant difference. These are, for all intents and purposes, identical numbers, okay? So this tells us that these symptoms that these people were experiencing are just as likely to have occurred whether they got a vaccine or they got a shot with nothing in it. So it's probably not actually due to the vaccine and probably more likely due to nerves. The second row that we're going to look at is what we call a solicited injection site reaction within 7 days. So what this means is that the researchers, they solicited a response. They reached out to people and asked them for a response. They said, within the first 7 days, how did your arm feel? Of the group that received the vaccine, who they solicited, which was about 4,000 or 4,100 people, about 78.6% of them said that their arm hurt. In the placebo group, it was approximately 12.8%. So this is going to tell me, as someone who was looking to receive the vaccine, two different things. The first is that I have more than a 75% chance, approximately, of experiencing some soreness in my arm after dose 1 and after dose 2. In the placebo group, you can expect, in a given population of about 4,000 people, you can expect that their arm is going to hurt because they got a needle in their arm, even though there was nothing technically in the shot. This tells me that I have a pretty good chance of experiencing soreness in my arm after both doses of the vaccine in that first 7 days. And if I do experience soreness, that it's actually because of what's in the shot and not just because I got a needle in my arm. The third one we're going to look at is the solicited systemic adverse event within 7 days. What this means is that they reached out to people and they said, how did the rest of your body feel? And after dose 1, about 60% of the respondents said that they experienced some sort of symptom. In the placebo, about 47% said that they experienced a symptom. This tells me, yes, I would say that this is statistically significant, although I haven't run the statistics myself, but this tells me that I've got a decent chance of experiencing some kind of symptom after dose 1 of the vaccine. But if I do experience it, it might be because of the vaccine or it might not be because of the vaccine. Some people are experiencing symptoms. Almost 50% of them in the placebo group experienced some kind of adverse event after dose 1. But after dose 2, things get a little bit interesting. After dose 2, it's closer to 70% of the respondents who experienced some sort of symptom in their body. But in the placebo group, it was only about 33.8%. So this tells me that I'm more likely after dose 2 than I was after dose 1 to experience a symptom in the first place. But further, if I do experience a symptom, it's probably because of what's in the vaccine and not because of some other external or confounding factor. What are these adverse events? The first one that we're going to go through is fever. 38 degrees Celsius is a little bit above our body's normal running body temperature. Our body normally runs at about 98.6 degrees Fahrenheit. 38 degrees is about 100 degrees Fahrenheit. After dose 1, they reached out to about 2,000 people and they said, did you experience a fever in the first 7 days after dose 1? About 3.7% of them responded with a yes, they did. Contrary to that, the placebo group, little less than 1% experienced a fever. So what this tells me is, sure, I'm not very likely to experience a fever, but if I do experience a fever, it's probably because of what's in the vaccine and not because of some other external factor. After dose 2, that number jumped up to 15.8%. So 15.8%, that tells me that after dose 2, I am more likely than I was after dose 1 to experience some kind of fever. Further, if you look at the placebo group for dose 2, it's about 0.5% who experienced a fever. So this tells me I'm still not very likely, but if I do have a fever, it is very likely because of my second dose of vaccine rather than because of some sort of external environmental factor. Next, we have a couple of other adverse events. So we've got fatigue. We're going to just look at any fatigue. After dose 1 of the vaccine, almost 50% experienced some sort of fatigue. In the placebo group, the number of people who experience fatigue is the percentage of people who experience just fatigue over the course of any 7-day period is approximately 33%. That's understandable. We're a pretty tired group of people. After dose 2, it went from 47% up to almost 60%. Whereas in the placebo group for dose 2, it's about 22.8%. So this tells me that if I experience fatigue after dose 2, it's probably because of the vaccine and not because of some sort of external environmental factor. Next, we'll look at headache. After dose 2 of the vaccine, you had about a 50% chance of experiencing a headache of any kind. And for the placebo, it's about a quarter of the people, about 24 or 25%. For chills, we've got 35.1% after dose 2, so greater than a third. But for the placebo, only about 3.8% of people experience chills on any given week. When we look at vomiting, 1.9% after dose 2 experience some kind of vomiting. So it's not a very likely chance that you'll experience vomiting of any kind. But with the dose 2 of the placebo, we had 1.2% who experienced vomiting. This tells me that if you experience vomiting after dose 2 of the vaccine, it's probably not because of the vaccine, because in a population of about 2,000 people, between 1 and 2% of them you can expect will experience some sort of vomiting. Yes, 1.9 is technically greater than 1.2, but I would not say that that is a statistically significant difference when you're looking at these sizes of populations. Me personally, when I read this, I said, okay, this is pretty close to what you would expect in a population, even if they didn't get the vaccine. This is probably not because of the vaccine. Then you look at diarrhea. You'll notice that these numbers are all pretty close to each other. There's a little bit more of a difference between these two, but not very much. The way that I interpret this so this is dose 1, this column's dose 1 of vaccine, dose 1 of placebo, dose 2 of vaccine, dose 2 of placebo. So dose 1 of vaccine, about 11% of them experienced some kind of diarrhea. Dose 1 of placebo experienced more diarrhea than dose 1 did. This tells me that if I were to experience diarrhea after getting the vaccine, I have the same chance of experiencing that diarrhea during that week as I did even if I didn't get the vaccine. Next, we have new or worsened muscle pain. This one, there's a significant difference. We've got 21%, so about a fifth of people experience some kind of muscle pain after dose 1 of vaccine. But in the general population, you can expect only about 10% of people to experience some kind of muscle pain, maybe because they were doing a workout or something like that. They might be experiencing delayed onset muscle soreness, for example. After dose 2 of the vaccine, it's about 37.3% who experience muscle pain or soreness. This tells me that after dose 2 of vaccine, I am more likely than dose 1 of vaccine to experience some kind of muscle pain. And further, in the general population, about 10% of them for any given week will experience some sort of muscle pain. We can expect 10% of people to experience muscle soreness in any given week, but in the people who actually got the vaccine, we can expect 37% of them to experience some kind of muscle soreness. Lastly, new or worsened joint pain. You have about 11% who experience some sort of joint pain after they received the vaccine versus 6%. It's kind of a gray area. I would say here, maybe it's because of the vaccine, maybe not. It's probably because of the vaccine. But then the difference here is a little bit more stark, right? So 6% and 5.2%, okay, we can expect about 5 to 6% of people over the course of a week are going to experience some sort of joint pain. But in the vaccine group for dose 2, about a fifth of them experience some type of new or worsened joint pain. This tells me If you experience this symptom, it's probably because of what's in the vaccine and not because that's just what's expected in the population. So one last thing before we wrap up the safety portion, I want to talk a little bit about allergies. Allergic reactions, they are rare, but they're not impossible. It's a risk with any vaccine, anything that you're getting injected with, there is a possibility that you could have an allergic reaction to it. The severity of the allergic reaction depends on the individual. The chance of an allergic reaction also depends on the individual. If you experienced a vaccine reaction to the anthrax shot, there is a possibility that you could experience an allergic reaction to the Pfizer-BioNTech vaccine. They looked at the ingredients of the anthrax vaccine and they compared it to some of the ingredients in this Pfizer-BioNTech vaccine, and there was either a similar ingredient or the same ingredient in both of these vaccines. If you have ever had an allergic reaction of any kind, make sure that when you go to get this vaccine, you notify the medical staff on site that you have had an allergy. You'll also have to fill out a form that says that you have had an allergic reaction to something, and it will ask what that something was. So you will have to put down that you have had an allergic reaction before, and then make sure that the people there have not only the knowledge of how to treat allergic reactions, but also have the equipment on site to treat an allergic reaction should it occur. Again, allergic reactions are extremely rare. During the week of December 14th to the 23rd, there were 21 cases of anaphylaxis in the first approximately 2 million doses of the first dose of the Pfizer-BioNTech vaccine. What this means is that you have about a 1 in 90,000 chance of experiencing anaphylaxis. So very small chance of this happening. For the people who experienced an allergic reaction, 71% of them occurred within that first 15 minutes of vaccination. That's why when you get the vaccine, they're going to go tell you to sit down and wait for 15 minutes. If you want to feel more comfortable, then you can wait 30 minutes. Now we're going to talk about the efficacy of the Pfizer-BioNTech COVID-19 vaccines. We have to talk about how many people actually completed this trial. There were just shy of 1,900 people who were slated to receive 2 doses of vaccine and just shy of 19,000 people who were slated to receive 2 doses of placebo. So we had over 37,000 people who were initially signed up to be vaccinated with either vaccine or placebo. The number of people who actually completed both doses of vaccine or both doses of placebo were 98.2% for the vaccine and 98.1% for the placebo. There's a couple of reasons why people don't finish vaccine trials. Some of them withdraw from the study, and when they withdraw, could be because they might have experienced an adverse event. That's what happened here. There was an adverse event that for 8 people who were in the vaccine group said was too much for them and they didn't want to continue with the drug trial. Then in the placebo group, you have 5 people. So statistically, these 2 numbers are identical. Further, you have 2 people who were slated to receive 2 doses of vaccine who died during the trial. You have 4 people who were slated to receive 2 doses of placebo who died. Now, if I were a sneaky statistician and I wanted to scare people, I could say this placebo is killing people. It killed twice as many people as the vaccine did. But the reality of the situation is rather that we can expect in a population of roughly almost 19,000 people that we're watching over the course of several months, we can expect that approximately 4 of them are going to die, maybe from car accidents, maybe from falling and slipping on some ice, maybe from a heart attack. There are a lot of ways that people can die. It is expected that a couple of people will pass away during the trial just because there are a lot of ways that people can die in real life. The fact that we had 2 people in the treatment group who passed away, statistically, these numbers are identical. Whether you got the vaccine or you got the placebo, you had about the same chance of passing away during the time that this vaccine trial was occurring, which was a couple of months. So what we're very interested in is this cumulative incidence curves for first COVID-19 occurrence after dose 1. What this graph is showing is new cases of COVID with respect to time. That's an oversimplified way of explaining this. So essentially what this means is that as time goes on, if these curves are going upwards, that means that there's more cases of COVID in that population. So the red line, or the red dots, those represent the people who received the placebo, and the blue dots, those are the ones who received the actual vaccine. So at day 0, they get dose 1, and then at day 21, they get dose 2. Now, what's interesting is that at about day 14, you'll notice that there's a split in the populations, right? If you couldn't see the whole rest of this graph, if you could only see up to here, you could say that maybe this split in the data isn't actually because of any real effect. Maybe it's just some randomization in the data. But you'll notice that from day 0 right up until about day 14, both the vaccine population and both the vaccine sample and the placebo sample of people are catching COVID, catching the disease at about the same rate. So as time goes on, they're both catching COVID at about the same linear rate. But then at day 14, there's that split. So that split, as you'll start to notice, becomes more apparent when we get to day 21, which is when they received their second dose of either placebo or vaccine. And the split is especially apparent that it is actually truly something significant, a true trend, once we get to day 28, which is 7 days after they received their second dose of vaccine. So as time goes on, and this is, the number of days, this goes out to day 119. As time goes on, the people who received the vaccine, they do not continue to catch COVID at the same rate. The way that we figure out how effective a vaccine is we split the population into a placebo group and a treatment group. And then we look at the number of COVID cases in the placebo group, and we compare it to the number of COVID cases in the treatment group. In the placebo group, people continued to catch COVID, but in the treatment group, people were not catching COVID like they were if they didn't get the vaccine. This tells us that this vaccine is effective. So how effective is it? 7 days after dose 2, it was 95% effective. Then what they wanted to know is how the data might look different if you look at different demographic groups. They looked at different ethnicities, they looked at different ages, and they looked at different genders. And no matter how they looked at the data, if they were only looking at one particular demographic group, it was always at least 93% effective. In some cases, it was greater than 95% effective. In some cases, less than 95% effective. But no matter how they looked at the data, no matter which subgroup they were looking at, it was always at least 93% effective, always. So this is really powerful. On average, it was 95% effective at preventing future incidents of COVID cases. Further, it was 100% effective at preventing death due to COVID infection, which is something that is also very powerful. So this vaccine, it's safe and it's effective. Now we're going to go through a couple of common COVID-19 vaccine myths. The first is that it was developed too quickly, and so it can't be safe or effective. But the reality is that there are lots of reasons why scientists were able to develop the vaccine so quickly and they didn't reduce its safety or effectiveness. The first is that the scientists were not starting from scratch. Even though the novel coronavirus is new, scientists have been studying the family of coronaviruses for literal decades. There was a scare back in 2001 in the media, and it was because of the severe acute respiratory syndrome, or SARS for short. It got a lot of attention, and SARS is caused by a coronavirus, similar to this coronavirus. Further, a couple of years later, MERS, Middle Eastern Respiratory Syndrome, it came to light in the media. It is also caused by a coronavirus. So while we were studying the family of coronaviruses before 2001, once 2001 came around, we really started paying more attention to coronaviruses and we started funding more research. So because we were studying the family of coronaviruses for decades, we had a lot of the foundational understanding of how these viruses work, and we were able to get a head start on the research that needed to be done before we made the vaccine. Next is coronavirus has affected every single continent on the planet, which meant that the entire world came together and we received unprecedented worldwide collaboration. Now, with collaboration, worldwide collaboration, not only do you get the brightest minds making this their top priority, but also you get lots of money. It's not that money makes research happen faster, but rather the lack of money will prevent research from getting done. The fact that they didn't have any shortage of funding really helped prevent this vaccine development from being hindered due to cost. Next, a lot of times it's very difficult to recruit people to participate in vaccine trials simply because it's risky, it's scary, and people don't want to risk their lives. But in this case, because so many people were affected by the pandemic, we had no shortage of people who wanted to help. Lastly, scientists could run some trials simultaneously rather than sequentially, as we showed in a previous slide, and that also saved a great deal of time. The next myth that we have is that the COVID vaccines alter your DNA. And the reality is that they don't, and here's why. This is a simplified diagram of our human cell, and you'll recognize inside you have an organelle called the mitochondria, which is the powerhouse of the cell, right? So the mitochondria is an example of an organelle, and there are other organelles that exist inside of your human cell. But one of the parts of the cell is called the nucleus, and inside the nucleus, that's where your DNA hangs out. But when you get the messenger RNA vaccine, it gets carried across the membrane and it goes into the cytoplasm, which is this watery, salty, grayish purple matrix out here. It gets translated from messenger RNA into proteins in the ribosomes of the endoplasmic reticulum. The messenger RNA never goes inside of the nucleus. And even if it did, the messenger RNA itself is just genetic material. It doesn't have any machinery of its own. Messenger RNA, if it's inside of the nucleus, gets shipped out of the nucleus to go and get translated into proteins. So there's no way that the messenger RNA is going to mess with your DNA. The next myth that we have is that the COVID-19 vaccine is just a way to microchip people. The reality is that there's no microchip in the vaccine. We know what the ingredients are. And the reason why we know what the ingredients are is because one of the conditions to get emergency use authorization from the Food and Drug Administration is that you have to provide a list of all of the ingredients that goes into your drugs. And that is publicly available. So we can go and look up the paper and see all of the ingredients that are in the Pfizer and Moderna vaccines. Anything that gets emergency use authorization, you can look up the ingredients yourself. So we already know what the messenger RNA does. It's the blueprint just for the spike protein. We know that the lipids transport the messenger RNA across the cell membranes. We know that the pH buffers and salts make it a stable solution for the lipids, not too acidic, not too basic, and the salts make it more comfortable to administer. And we know that it has sugar in it, which stabilizes it for freezing and thawing. None of these are to make a microchip. The last myth is that the COVID vaccines make your body make the virus. Here's why that's impossible. The messenger RNA is the blueprint to make just one piece of the virus, the spike. It doesn't code for any other components of the coronavirus. So we're going to use an analogy. So think of the coronavirus as this whole car. And think of the spike protein as just this little piece, this little side mirror, just that part of the car. The messenger RNA in the vaccine is a blueprint just to make this mirror, just to make the spike, just to make this piece of it. So no matter how many copies you make of that mirror or of the spike, even if you put all of them together, you still don't have a car and you still don't have a virus. So your body is not going to be making the virus. It's just going to be making one single piece of the virus. Now we're going to talk about how to sort through misinformation on the internet and out in conversations. When you are considering a news article or a meme or a story on TV, you always want to check and see if the headline is alarmist or sensational, or if it looks like it's attacking a specific group, whether it's a political group or a religious group or anything else. Usually, reputable sources don't really rely on scare tactics. They don't try to make anyone angry, and they don't make an effort to attack a particular group. It doesn't mean that that particular article isn't reputable. It's just that it's pushing one narrative of a story instead of the full story objectively. You want to utilize objective fact-checking websites such as Snopes, and if you don't like Snopes, you can Google objective fact-checking website and choose another one. You're always going to want to consider the source. So check for a media bias by searching for that news channel or journal, followed by bias. Lastly, always check to see if the article is citing peer-reviewed scientific journals. And then the journal itself, you want to make sure that it doesn't have too much of a bias. So you can search for the name of the journal, followed with reliability or bias. A lot of times people are not going to do all of these steps because it's a lot of steps. And they're especially not going to follow those steps if they read something that already aligns with a bias that they already have. We call this a confirmation bias. Because of that, people in general are far more likely to click share on their social media rather than taking all of these steps to check to see if the source is reputable. And for that reason, because people have this propensity to just click share whenever they see something that confirms their bias, you always want to be extremely wary of any articles that are shared on social media. I would like to thank all of the essential workers, all of the first responders, and the medical personnel who are risking their health and their safety to provide essential services and fight the spread of COVID. I want to thank all of the scientists and the volunteers who aided in the research and development of the vaccines. I want to thank the content creators, i.e. PBS, and thank you to all of you for watching this brief and paying attention. I feel that the best way that we can fight misinformation is to arm ourselves with the facts, and that was the goal of this brief, is to provide that information so that people can make those logic-based decisions rather than fear-based decisions. Thank you, and see you around.